Today, we're going to define and identify the characteristics and components of the innate immune system. And we're going to develop an understanding of the various innate mechanisms of recognition and response to infection. When people talk about the immune system, they're usually talking about white blood cells and antibodies. And it is true that our WBCs and the antibodies that our B lymphocytes or B cells produce are important. However, there is much more to the immune system than that, as we're going to see today. I would like to start by looking at the cells in our blood. All of our blood cells, whether anucleated RBCs that transport oxygen or our white blood cells that are involved in fighting infections, are derived from hematopoietic stem cells. There are a number of cells that are involved in protecting us from infection. The first four on this table, the helper T cell, the cytotoxic T cell, the B cell, and the plasma cell, are going to be covered when we go over the adaptive immune system. The rest of these are related to innate immunity, and this is with the understanding that there is an interaction that takes place between the innate and adaptive immune systems, and that will become clearer as we go through this entire unit. And even though cells like the eosinophil, which is involved in the elimination of parasites, the basophil, which is involved in promoting the inflammatory response, the mast cell, which is also involved in the inflammatory response and is implicated in allergic responses as well, and the dendritic cell, which is an antigen-presenting cell that also is a phagocyte in some ways. Even though all of these cells are very important in the immune system, today we're going to focus primarily on the natural killer cell, the monocyte, the macrophage, which is derived from the monocyte, and the neutrophil. Now, before we start talking about mechanisms of the immune system, let's go ahead and take a look at the organs that comprise the immune system. So in humans, prenatally, hematopoiesis, or the formation of blood cells, occurs in the yolk sac, then in the liver, and then lastly in the bone marrow. And it's in the bone marrow where hematopoiesis in adults occurs. And then some of these cells that are formed in the bone marrow mature in other lymphatic tissues, such as the thymus, which provides an environment for the development of T lymphocytes. Now, lymph nodes are small bean-shaped structures that are part of the body's immune system that filter substances that travel through the lymphatic fluid. And they contain lymphocytes that monitor this lymphatic fluid for foreign substances. The spleen filters the blood as part of the immune system. Old red blood cells are recycled in the spleen, platelets are stored there, and white blood cells are present and monitor for the presence of foreign substances. And then we have the mucosa-associated lymphoid tissue, or MALT. And this includes the tonsils, the appendix, and a large number of other follicles that are located in the connective tissue under mucosal surfaces. And any antigens that pass through the mucosa will encounter lymphocytes such as B cells and T cells, and also macrophages, which could keep the pathogens from entering the body. Our body's defenses can be separated into two subcategories. However, interaction does take place between components of both systems to produce an integrated response to infection. Our innate immunity, or non-adaptive resistance to infection, involves both physical and chemical barriers to infection. In addition, there are responses to infection that involve molecular agents and innate immune cells. But the innate response to infection doesn't change over time. These mechanisms never get better at responding to infection. That isn't the case for the adaptive immune response. Now, the innate response to infection is very rapid, and the adaptive immune system is much slower in responding to infection. But the response is much more specific and targeted to the particular pathogen that is causing the infection. Additionally, our adaptive immune system becomes conditioned. It acquires the ability to respond more readily to a second instance of infection by the same pathogen. Of course, the best defense to infection is one that keeps infection from ever taking place. The skin is a nearly impenetrable physical barrier to infection. And at the mucosal surfaces of the body, where our epithelial is more permeable to facilitate things like gas exchange and nutrient absorption, mucus traps pathogens and impedes their movement into the body. And finally, chemicals like lysozyme, which digests peptidoglycan, a component of bacterial cell walls, and stomach acids, which can kill microbes, and digestive enzymes found in the intestines that can digest microbes, all add to our first line of defense that keeps infection from ever taking place. And additionally, we can consider our microbiota to be another innate barrier to infection. We have more than 1,000 microbial species that call our gut home. And we've evolved alongside these commensal species, which enhance nutrition and additionally prevent infection by crowding out disease-causing microorganisms. And if a pathogen gets past our barriers and infects our body, our next line of defense is triggered to rapidly eradicate the infection. This is carried out by soluble antimicrobial proteins that are present in our blood and lymphatic fluid, such as complement proteins and defensins. And also, finally, our first-line cellular defenses, like neutrophils and macrophages, which engulf and digest pathogens in a process called phagocytosis, and natural killer cells, which seek out our own virally infected cells and trigger their destruction via apoptosis, or programmed cell death. So what is a pathogen? A pathogen is a bacteria, a virus, a fungi, or a parasite that causes a disease. Now, some pathogens are merely opportunistic in nature in that they can typically colonize our bodies and are not pathogenic. However, once they get into the wrong area, such as E. coli making its way into the urethra, or are able to proliferate uncontrollably, such as Clostridium difficile and its expansion in the colon that takes place after prolonged antibiotic therapy. Now, phagocytosis is a process that macrophages and neutrophils carry out. What they do is they engulf and digest pathogens. Pictured here on this diagram, we see a bacteria being bound by receptors on the phagocyte, which the phagocyte would be the cell that's carrying out phagocytosis. And that bacteria is engulfed in a type of endocytosis and encased in a phagosome. Following that, a lysosome, which is an organelle that contains digestive enzymes, fuses with the phagosome, forming the phagolysosome. Then the digestive enzymes that are introduced to the phagolysosome digest the bacteria. And then via exocytosis, the debris is released from the cell. Natural killer cells seek out and destroy host cells that have been infected by a virus. So when a natural killer cell detects a virally infected cell, they attach to it and exocytose perforin and granzyme. And perforin polymerizes in the plasma membrane of the target cell, forming a pore that allows granzyme to enter the virally infected cell. Granzyme then triggers the apoptosis process, which results in cell death. One of the keys to apoptosis is that it doesn't cause the infected cell to rupture, which would result in releasing its contents and the viruses that are being formed inside of it. Instead, this apoptosis process is very orderly. The contents of the cell actually are contained in vacuoles, and a macrophage is seen here coming by and cleaning up the mess. Now, the complement system is one of the molecular responses to infection. It consists of over 30 different proteins that are in an inactive state in the blood. One of the ways that complement is activated is by interacting with a molecule called mannose-binding lectin. When MBL binds to a pathogen and encounters certain complement proteins, it activates those complement proteins. And one of the outcomes of complement activation is the formation of a pore that in gram-negative bacteria and enveloped viruses can result in the lysis of the pathogen. And this pore is called a membrane attack complex. So the way that the innate immune system actually recognizes infection is by binding pathogen-associated molecular patterns. These are structures that are either found on the exterior of pathogens or molecules released by the pathogens. These are chemical structures such as lipopolysaccharide or peptidoglycan from bacteria, or things like viral DNA and RNA. Our cells have pathogen recognition receptors, such as toll-like receptors, that bind to these pathogen structures, and this binding is what elicits a response. We also have soluble PRRs, meaning that they aren't attached to a cell, but are in the blood or the lymphatic fluid, like mannose-binding lectin, that also, once they bind these pathogenic structures, elicit a response. Now, this diagram or flow chart from the textbook illustrates how these interactions take place and the outcomes that result from them. So via the binding of these pathogen-associated molecular patterns by pattern recognition receptors, the innate immune system reacts to the presence of pathogens and attempts to neutralize the pathogens. So just to go through a few of the processes that are depicted on this diagram, bacteria and fungal components that are bound by mannose-binding lectin results in the activation of complement proteins. One of the outcomes that we just spoke about is the formation of the membrane attacked complex, which targets gram-negative bacteria and enveloped viruses. Another thing that can take place due to complement activation is the formation of C3b. C3b is an active form of complement protein C3. And what C3b does is it actually binds to microbes and allows circulating phagocytes to more easily phagocytose the pathogen. Similarly, epithelial cells can respond to these bacterial and fungal components because they possess toll-like receptors on the outside of them. And when these epithelial cells bind these components at their toll-like receptor, that elicits the production of chemokines, which are chemicals that attract circulating phagocytes and natural killer cells to the binding site. Additionally, the epithelial cells will release antimicrobial peptides that target the pathogens. The same response can be triggered in epithelial cells by viral RNA, DNA, and proteins. Now, if macrophages encounter bacterial or fungal components or viral RNA, DNA, and proteins and bind it to their toll-like receptor, they will also produce chemokines that attract other phagocytes and natural killer cells to the area. But in addition, they will produce cytokines which ramp up the responses carried out by circulating phagocytes and natural killer cells. Another thing that can take place, of course, is that when a virus infects a cell, that cell presents different structures on its exterior due to the viral infection. And that can be directly detected by the natural killer cells. Then they can destroy the virus-infected cells in the process that we just talked about, using perforin and granzyme. Now, the inflammatory response is another response by the innate immune system, and we can use that to illustrate the ability of neutrophils and macrophages to move from the blood into the tissue. So let's say a nail covered in bacteria penetrates the skin. Due to the tissue damage, chemicals are released at the site of the infection. These chemicals, what they do is they attract neutrophils and monocytes to the area. They increase blood flow by causing dilation of the local blood vessels, and they also increase the permeability of these blood vessels, which allows the neutrophils and the monocytes to move into the tissue from the blood via a process called diapedesis. The monocytes, once they're in that tissue, differentiate into macrophages. Then the neutrophils and the macrophages proceed by phagocytosing the invading bacteria. So the inflammatory response is actually a response to infection that's triggered by the innate immune system that aids in neutralizing the pathogens. So today we defined some characteristics of innate immunity, the fact that it's rapid acting, it's our first line of defense, and it doesn't adapt. We looked at some of the components of innate immunity, such as the physical and chemical barriers, and also the soluble factors and cellular defenses that are used to neutralize infection. And then we talked about some of the recognition and response mechanisms to infection, such as PAMPs and PARRs, phagocytosis, and apoptosis triggered by natural killer cells. And one of the things to keep in mind with the innate immune system is that infections are taking place all the time. When the innate immune system is able to combat these infections, we typically are not even aware that they've taken place. So in the next lecture, what we're going to talk about is the adaptive immune system, which is slower to react, but a much more powerful response. And it's what takes place when the innate immune system is not a sufficient response to eradicate an infection.